Hi everybody, sorry I'm not there today, but let's go ahead and continue on where you guys were taking notes last night. And so last night we talked about Beadle and Tatum and how they discovered that each gene codes for a different enzyme. And so just so you guys know, really we know now that it's really one gene codes for one protein. Okay, so every single type of protein we have of all the different shapes, whether that be a surface protein, a receptor protein, enzymes, structural proteins, flagella proteins, any of these proteins that we would ever need to make are coded for in our genes. Okay, so our DNA is very long and it's composed of many genes. So you can think of a gene as a recipe for a protein. Now, to get more complex, we found out that sometimes proteins have several subunits. So you'll remember like hemoglobin, for example, has four different subunits like this, with iron in the middle. Okay, and so these four proteins come together and they may be coded for by four different genes. Okay, so now they've even further classified it as one gene, one polypeptide. All right, so a polypeptide, you'll remember, is a bunch of amino acids together. Those will then fold up into their secondary and tertiary structure, and oftentimes will take multiple subunits to form a quaternary structure. So it'll look something like this. All right, and this could be composed of four different genes. Okay, now let's start with step one, which is transcription. So protein synthesis, remember, it's going to be happening on the ribosomes, generally speaking. It happens anytime any cell needs any protein. So you are doing this currently all over every single one of your cells. Okay? And the first thing we need to do is take a look at the DNA and find out where the recipe is. Where is the gene for the protein that we currently need? All right, so wherever that gene is, we will unzip that piece of DNA and we will make a copy of it. And that copy is called the mRNA transcript. Okay, so the process of copying a gene is called transcription. It kind of sounds like a scribe copies things, right? In the old days before copy machines, you would hire a scribe to copy books. Now we apply that same term here, so we're copying. Okay, now we're looking actually at a prokaryotic cell in here. So you see there's no nucleus where the DNA is, right? And if we were actually to make this a little bit more accurate, this would actually be in a shape of a circle. Okay, so they have one big circular chromosome here. So they've got a ribosome that comes in and sandwiches itself around the mRNA. And the ribosome is just going to go along that mRNA, read it, and start to put together our amino acids. So each one of these little balls is an amino acid. Okay. Now, if we actually look at eukaryotic processes, now we see we have a nucleus, okay? So you should know that for eukaryotes, transcription occurs in the nucleus, because duh, that's where the DNA is. So this is just one of our chromosomes. We know we have 46 of these chromosomes, okay? Now, when we look at the gene that we want to copy, the first thing that's going to happen is we're just going to make a straight copy of it, okay? Now, let me blow this up for you a little bit. This step where we actually make mRNA, let's just go over mRNA a little bit, okay? So our DNA is going to unzip. So imagine this is the half of the DNA that's going to get copied, okay? The other half is down here. Okay, so this is the unzipped DNA. Let's say, for example, we have C, C, T, A, T, T, G, G. Okay, take a minute. What would the mRNA copy of this read? Okay, if we're actually going to make the mRNA here, how would we base pair? Okay, so if this was the RNA, we would come in, C matches with G, and RNA is single-stranded, and instead of deoxyribose, it's going to be ribose, okay, on the backbone. So the sugar phosphate backbone is a different sugar. So we're going to have G, A, and here's another difference. Remember, RNA has U, so A is going to pair with U, A, C. Okay, so this copy down here is what's called our pre-mRNA. Now, we go through a bit of RNA processing. So that means we're going to add two things here, and I'll go over that on the next slide. We're going to add these caps, okay? And notice the RNA gets shorter. We're going to go through what's called RNA processing. So that means we have to finalize the mRNA so it's ready to go for translation here, okay? And once it leaves, it's going to go through the nuclear pore out to the ribosome, and we'll translate that message into the actual polypeptide. Okay, so we already said transcription is making an RNA copy of a DNA gene. So let's take a look. This process, let's go back. Remember, we're only going to copy a certain section of DNA. We don't need the entire recipe book all the time. This is not a cell that does everything. It's a specialized cell. So we're only going to make certain proteins here. So how does the cell know which part of the DNA to copy? It needs a signal, right? All right. So there's a number of things, okay? So here's the thing. Here's our DNA here. You remember it's 5 to 3 and everything, okay? And so let's say this blue part right here is the actual gene, okay? So you'll notice there's a start point here and this section in the beginning called a promoter. All right, the promoter is marked by what's called the ta-ta box, okay? This is just a series, kind of like it sounds, of T's and A's. And so this is going to basically be sort of a landing spot for all the proteins and factors required to copy a gene. All right, so these things right here, these are transcription factors. Remember how we talked about in cell signaling, one cell will release a signal, right? And that signal is going to get picked up by a target cell, okay? So let's say this cell needs to respond, okay, in some way. So once the signal binds to the receptor, remember that's going to change the shape of the receptor, and that's going to activate a number of enzymes, okay? Now, the products of those enzymes are the transcription factors. Remember we said that once a cell is communicated with, it's possible that that communication could result in a difference in the way the DNA is used. So gene expression changes based on what's going on outside of the cell, okay? So, for example, if the pituitary gland needs to make a hormone, it's going to get a signal, right? So these transcription factors are basically proteins that are made inside the cell as a result of that communication. So if the communication happens, the transcription factors get made, and they're going to come over here and they're going to match, okay? They are going to chemically bind with a particular gene, okay? So these transcription factors exactly match the chemical properties of that promoter right there, okay? Now, if those transcription factors are present, they're going to bind, and this guy right here, RNA polymerase, is going to be able to bind, okay? It's going to be able to initiate, all right? And once this enzyme can come in and initiate, then we're going to start making our mRNA transcript down here, okay? So we don't make mRNA, we don't copy a gene until the transcription factors are present, because why would we want to make a protein we don't need? So the transcription factors, they're kind of like our signal, and we wait for them to bind before the RNA polymerase comes in, okay? Okay, so I'm going to show you a quick animation of this. Let's go here. Okay. All right, there's our promoter. You notice the ta-ta region right here. Okay, we're going to unzip, and the RNA polymerase is going to go ahead and base pair, until we reach what's called the termination sequence. Okay? All right, now this is going to be our mRNA transcript, and let's go back to what's going to happen next. Okay, now the next thing that's going to happen is we have to process that RNA. So let's see what happens there. Okay.
Most eukaryotic genes are composed of numerous short coding sequences called exons, embedded within stretches of non-coding sequences called introns. The initial messenger RNA molecule, or primary transcript copied from a gene by RNA polymerase, is a faithful copy of the entire gene, including introns as well as exons. Before the primary transcript is translated, the introns are removed by a process called RNA processing, or RNA splicing. Particles composed of proteins and a special type of RNA called small nuclear RNA, or snRNA, play a role in RNA splicing. One kind of small nuclear ribonucleoprotein, snRNP, contains snRNA that can bind to the 5' end of an intron by forming base pairs with complementary sequences on the intron. A different snRNP binds to the 3' end of the intron. Additional introns interact, causing the intron to form a loop, thereby bringing the two ends of the intron together. The large complex of snRNPs, called a spliceosome, then excises the intron and the exons are joined together. The snRNPs are then released.
Okay, let's talk about that in English, okay? Let's go back to this. So let me get my pen here. Okay. All right. So basically what's going on is we have our DNA, right? We're just going to copy a little piece of that, right? We're going to make a single-stranded copy mRNA, right? And so we're going to, like it said in the video, pieces of this get cut out, all right? And this is how they discovered this. They started to count up all the proteins that a cell makes, okay? Let's say they counted up that the cell makes 1,000 proteins, okay? Any cell in a given organism, the organism can make 1,000 proteins. So one would assume that there would be 1,000 genes, right? If we looked at the DNA, wouldn't that correspond to 1,000 genes based on the Beetle and Tatum stuff? They found out it's not that way. So there's not 1,000 genes, there's quite a bit less actually. If we counted up all the genes, there would be something more like 700 genes. So it's like, how do we make 1,000 proteins with only 700 genes? And it turns out it is called alternative splicing. Oops, splicing. Okay, I do this all the time in my kitchen, right? Let's say I look up a recipe for pizza, right? Maybe it has pepperonis on it. Okay, and then they tell me to put some mushrooms on it. Okay, got that. Some basil, right? Some cheese. Okay, here we go. I'm making our pizza. All right, but then they tell me to put olives. Gross. I don't want any olives. I'm going to splice that right out of the recipe, okay? And now I have a pizza that's a little bit different than what was supposed to come out, but it's just a little bit different, right? I just left out the 1 olives. No big deal, right? Well, that's what happens here. So as soon as this mRNA is copied, we splice out sections that we don't need, and we splice together the parts that we do, okay? Now, the parts that leave, okay, the parts that get cut out, these are called introns. It's a little counterintuitive the way it's named, but follow my train of thought here, okay? The parts that are actually spliced together are called exons. So the exons are going to make up the processed mRNA. So that's like the final recipe here. And this final recipe is going to exit. So if you think about it, this is all going on in the nucleus. The exons are able to exit the nucleus. And that transcript will go out to our ribosome and get made into the proper protein. Okay? So you see here, you could imagine that one day, these are the introns that are going to get cut out. Maybe the next day a different protein's needed. So what used to be an exon might be an intron, okay? So maybe my friend's coming over that likes olives, and she just likes olive pizza. So I'm going to cut out all the mushrooms and all the other stuff, okay? So depending on what the needs are, we can alternatively splice together these genes. Or, I'm sorry, these nucleotides. Excuse me. Okay, so we'll talk more about that next time in class. Hope that makes